First part of excretory system, we dealt with the parts of excretory system of mammals and the process of urine formation along with its regulation. In this part, we shall deal with the process of urea formation in the liver and its related disorders in mammals. Excretion refers to the elimination of waste products from the body in order to maintain homeostasis and protect the body from their toxicity. The waste products include nitrogenous materials, carbon dioxide, pigments, excess water, inorganic salts, vitamins and hormones. Among these wastes, the nitrogenous waste materials are removed in the urine. The rest are removed by sweat glands, liver and lungs. Both excretion and osmoregulation are helpful in maintaining homeostasis or constant favorable internal environment of the body. In vertebrates, these functions are performed by kidneys and their associated structures in urinary system. Excretory products and their types. The excretory products are of different types: volatile products and non-volatile products. The volatile products, such wastes are evaporated immediately from the body and include water vapor, carbon dioxide, ammonia, ketone bodies, etc. The non-volatile wastes are removed very slowly in aqueous form, which includes nitrogenous products such as urea, uric acid, sulfates and phosphate ions, etc. The nitrogenous wastes are the principal excretory product and are mainly formed as end products of protein metabolism. The principal nitrogenous waste products vary in different animals depending on their evolutionary history and their habitat. There are three main nitrogen containing waste materials excreted by animals, which are ammonia, uric acid and urea. Depending upon the form in which these waste is excreted from the body, the organisms are grouped into three categories accordingly, such as ammonotelic, uricotelic, ureotelic and aminotelic. Ammonotelic animals. Their nitrogenous waste is in the form of ammonia. So, this phenomenon of removing waste as ammonia is known as ammonotelism. It is formed by oxidative deamination of amino acids. It is highly toxic except in dilute conditions and should be eliminated from the body quickly. Large amount of water is required for dissolving and removing ammonia. All marine and aquatic animals, bony fishes, earthworm, etc. belong to this category. Uricotelic animals. Their nitrogenous waste is uric acid and urates. So, this phenomenon of removing waste in the form of uric acid is known as uricotelism. Uric acid is formed from ammonia by degradation of proteins mostly in liver and to some extent in kidneys. Uric acid molecule is more complex and requires more energy. It is much less toxic than both ammonia and urea. It is almost insoluble and is eliminated in almost solid state, thus saving water. All terrestrial animals like insects, reptiles like lizards, snakes, turtles, etc. and birds are uricotelic animals. Ureotelic animals. Their nitrogenous waste is mainly in the form of urea, and this phenomenon of removing waste in the form of urea is known as ureotelism. Urea can be stored in the body for considerable periods of time, and it is least toxic in nature. It is more complex in nature and requires energy for its production in liver by a cyclic process called urea cycle or ornithine cycle or Krebs-Henseleit cycle. It is observed in terrestrial animals with limited access to water such as adult amphibians, reptiles and mammals including man. Ammonia is converted to urea in liver. The last is aminotelic animals. Their nitrogenous waste is in the form of amino acids. So, this phenomenon of removing waste as amino acid is known as aminotelism. In certain mollusks and echinoderms, excess amino acids are excreted without any change. Besides these wastes, other wastes are also found, which includes guanine found in spiders, trimethylamine oxide in marine teleosts and bony fishes, hippuric acid. Another type of waste is formed from benzoic acid and glycine and is found in rabbits. Ornithuric acid is found in birds and is converted from ornithine. Creatine and creatinine are synthesized from arginine, glycine and methionine in higher mammals. Formation of urea by ornithine cycle or Krebs-Henseleit cycle. We shall now discuss the formation of urea in the liver cells. This cycle was proposed by Hans Krebs and Kurt Henseleit in 1932. Only the liver cells can convert ammonia, which is produced from protein metabolism, into urea by combining with carbon dioxide. This is a pathway that spans two cellular compartments. The urea cycle consists of 5 reactions, 2 mitochondrial and 3 cytosolic enzymes. 5 enzymes and 3 ATP molecules help in urea formation. 3 amino acids participate in the process. Urea contains 2 nitrogen atoms, one comes from ammonia and the other from aspartic acid. The cycle can be understood by the following steps. 1. Synthesis of carbamyl phosphate. Free amino acids are released from dietary or intracellular proteins. The alpha amino nitrogen is removed by deamination to release ammonia. This ammonia is removed from the circulation by liver to form urea. The first step of urea cycle begins in the mitochondrial matrix. The enzyme carbamyl phosphate synthetase 1 combined with ammonia, bicarbonate, water and 2 molecules of ATP, that is adenosine triphosphate, in presence of magnesium ion to produce carbamyl phosphate. The second step, synthesis of citrulline. In the second reaction, carbamyl phosphate enters into the urea cycle and donates its carbamyl group to ornithine to form citrulline and release inorganic phosphate. In a reaction catalyzed by ornithine transcarbamylase, the citrulline is released from the mitochondrion into the cytosol. The subsequent reactions occur in the cytosol. Citrulline is transported from the matrix of the mitochondria to the cytosol by a transporter called citrulline transporter. The third step, synthesis of arginosuccinic acid. In the third step, second amino group is added in the cycle in the form of aspartic acid, which joins with citrulline to form arginosuccinate. The reaction is catalyzed by arginosuccinate synthetase in presence of adenosine triphosphate and magnesium ions. The fourth step, cleavage of arginosuccinic acid to arginine and fumaric acid. In the next step of urea cycle, arginosuccinate lyase reversibly cleaves arginosuccinate to produce arginine and fumarate. Arginine contains 4 atoms of nitrogen. The fumarate then enters the mitochondrion of liver cells and enters the pool of citric acid cycle intermediates. The fifth step, cleavage of arginine to ornithine and urea. The last reaction of the urea cycle is catalyzed by cytosolic enzyme called arginase, which is present only in liver cells of all ureotelic organisms. This enzyme arginase hydrolyzes arginine to produce urea and ornithine, each containing 2 atoms of nitrogen. Ornithine is then transported back in the matrix of the mitochondria by ornithine transporter to complete the cycle and to initiate another round of the urea cycle. The urea thus formed is passed via the bloodstream to the kidneys and is excreted into the urine. A defect in any of the enzymes of the urea cycle leads to a toxic accumulation. The overview of the cycle is that amino acids get deaminated to release ammonia during amino acid catabolism. This amino acid combines with carbon dioxide and adenosine triphosphate inside the mitochondrial matrix to form carbamyl phosphate in presence of carbamyl phosphate synthetase 1. The carbamyl phosphate thus formed combines with ornithine to form citrulline by the ornithine transcarbamylase. Citrulline leaves the mitochondrial matrix and enters the cytosol to combine with aspartate and accept the ammonia group to form arginine and water. Arginine then decomposes to form urea and regenerate ornithine for reuse in the cycle in presence of arginase and water. Thus, in the complete ornithine cycle, 2 molecules of ammonia, 1 molecule of carbon dioxide, 3 ATP and 5 enzymes are required for formation of 1 molecule of urea. Advantages of urea as excretory waste. Now, let us see the advantages of urea as nitrogenous waste material in ureotelic animals. These advantages are: 1. It is almost neutral and far less toxic than ammonia. 2. It can accumulate in higher dose without adverse effect. 3. It can be excreted in a more concentrated form. Urea is also highly soluble in water and requires a good deal of water for its removal from the body. About 1 gram of urea requires 50 ml of water to dissolve. Hence, it is excreted as the main nitrogenous waste product by animals which can lose lot of water without adverse effect, like prawn, toad and man. Moreover, the concentration of urea in the blood can be tissue fluid, can be raised in animals like sharks and rays. Regulation of urea cycle. The rate of synthesis of the 4 urea cycle enzymes and carbamyl phosphate synthetase 1 in the liver is regulated by changes in demand for urea cycle activity. If the dietary intake is primarily proteins, much urea is formed. During prolonged starvation, breakdown of muscle proteins take place resulting in much urea formation. The activity of urea cycle is regulated at two levels, high and low enzyme concentration. The enzymes are synthesized at higher rates in animals during starvation, in very high protein diet. Enzymes are synthesized at low rates during well-fed animals with carbohydrate and fat diet, animals with protein-free diets. The deficiencies of urea cycle enzymes. Defects in many enzymes of a metabolic pathway can result in essentially identical signs and symptoms. All defects in the urea synthesis result in ammonia intoxication and becomes more severe when metabolic block occurs at the first and second step of reaction. Urea cycle disorders occur in both children and adults. The clinical symptoms include vomiting, avoidance of high protein foods, intermittent ataxia, irritability, lethargy and mental retardation. A blockage in any of the steps of the urea cycle has serious consequences as there is no alternative pathway for the synthesis of urea. Some of the defects of the urea system are hyperammonemia type 1. The deficiency of carbamyl phosphate synthetase 1 causes hyperammonemia, that is high concentration of ammonia. 2. Hyperammonia type 2. A deficiency of Oniton transcarbamylase enzyme produces X-linked recessive disorder caused by a number of different mutations in the Oniton transcarbamylase gene. Males are generally more seriously affected than females as they are heterozygotes. It is the most common urea cycle disorder, resulting in a mutated and ineffective form of the enzyme. The complications with this Oniton transcarbamylase autosomal recessive metabolic disorder may include mental retardation and developmental delay. Citrilemia, also known as citrullinuria. It is a rare disorder and is recessively inherited. It is caused by deficiency of arginosuccinate synthetase. This autosomal recessive metabolic disorder is unable to condense citrulline with aspartate, resulting in accumulation of citrulline in blood and its excretion in the urine. There are two types of citrullinemia. In type 1 citrullinemia usually becomes evident in the first few days of life. In type 2 citrullinemia, the signs and symptoms usually appear during adulthood and mainly affect the nervous system. The therapy includes specific supplementation with arginine for protein synthesis and for formation of creatine and ornithine. Ammonia encephalopathy. This occurs when there is increased concentration of ammonia in the blood and other biological fluids. Ammonia diffuses into cells across the blood-brain barrier and increases the synthesis of glutamate from alpha-ketoglutarate. Glutamate and ammonia elevates the glutamine levels, which leads directly to brain damage. It may affect the nervous system and cause bizarre behavior. Hyperarginemia. This defect is characterized by elevated levels of arginine in blood and cerebrospinal fluid and low erythrocyte level of arginase. There is a probable competition by arginine with lysine and cysteine for reabsorption in the renal tubule. A low protein diet lowers the plasma ammonia levels and abolishes urinary lysine cysteine urea. In case of infants born with total deficiency of one or more enzymes of the urea cycle, survival is seen only for few days. Interruption of the cycle at each point affect the nitrogen metabolism differently. Some of the intermediates can diffuse from hepatocytes and accumulate in the blood and pass into the urine. If symptoms are not detected early enough, severe mental retardation results, causing irreversible brain damage. Conclusion: different types of nitrogenous wastes are found in animals depending on their habitat. In humans, the waste is urea, which is produced in the liver by a process called Oniton cycle or Krebs-Henseleit cycle. Urea is the major metabolic end product of nitrogen catabolism in humans. The urea cycle is energetically expensive. The synthesis of 1 mole of urea requires 4 high energy phosphates. The synthesis of urea in liver is the major pathway of the removal of ammonium ions. The citric acid cycle and urea cycle both are linked together. The fumarate produced in the reaction of urea cycle in the cytosol by arginosuccinate lyase reaction is also an intermediate of the citric acid cycle. The metabolic disorders of urea biosynthesis are extremely rare.